Good morning, all students. Anyone tell me screen is visible or not? Roshan Yadav, Hitesh,
Yes, sir.
all of you got the marks of chemistry of the online November test? I already displayed the marks in the form of PDF on the your WhatsApp group, but by the order of the principal, again I deleted it because some student didn't pay the fees of the colleges. So I request all the students, those who didn't pay the fees or they didn't take the admission for the 12th standard, please take the admission. Because of these students the result will be not displayed. Result will be displayed by the exam department. They will separate the students, those who's paid the complete fees, those who do not pay the fees according to that. But I observe all the students written very nice papers. A paper was actually very easy. The reactions are very easy and the number of chapters are also less. So all the students are passed and the result, our result is 100%. After the order of examination department, again I will display it on the group. So we will start the next part. Yesterday we discussed about this remaining part of the carbohydrates, disaccharides. Once again, I will take a quick revision of these disaccharides, structure of this sucrose. This one. Yesterday we discussed about these disaccharides. Disaccharides means when these are hydrolyzed, they gives only 2 units of monosaccharides, and these two monosaccharides are joined together with oxygen atom. This is called as glycosidic linkage. This can be asked for the examination. What is meant by glycosidic linkage? These structures also we discuss about the structures. So this is the sucrose. It is prepared with one alpha D glucose unit and beta D glucose unit. Now you are familiar with what is meant by alpha and what is meant by this capital D. Alpha means again I will show this is a beta side. Beta side means this uh, see this is alpha side and this is a in case of alpha, this OH is below, and in case of this beta, this OH is above. Which OH? In case of glucose, we are considering the carbon number. Sorry, all these are the fructose. In case of fructose, we are considering carbon number 2, and in case of glucose, we are considering carbon number 1. See, this is a carbon number 1, OH is below, it is alpha, OH is above, it is a beta. See, that's why it is alpha D glucopyranose and this is a beta D glucopyranose. In case of glucose, we are considering this carbon number 1 as an anomeric carbon. And in case of fructose, we are considering this second carbon. Second carbon is a anomeric carbon. This is when OH is below, it is alpha, and OH is above, it is beta side. So observe this structure carefully. This is alpha D glucose. OH is below and this is beta D fructose, OH is above. This structure can be asked for 1 mark. So they remember this linkage. In case of sucrose, linkage is alpha beta 1,2 glycosidic linkage. This linkage, remember. And in case of maltose, Both the monosaccharides are same. This is also alpha D glucose. This is also alpha D glucose. So the linkage is 1,4 glycosidic linkage. Now, the structure of maltose is very easy. And in case of lactose, it is also called as milk sugar. See the linkage. This is 1,4. Actually, these galactose and glucose, these are structures are same. Sorry, the molecular formula is same, but only structures are different at carbon number 4. See, in case of carbon number 4, in case of galactose, see, this OH is above and H is below, but in case of glucose, H is above and OH is below. This is the only change between galactose and glucose. Both these are stereoisomers. It is also mentioned in the notes also. Galactose, glucose are the stereoisomers. They differ in their stereochemistry at carbon number 4 only. Otherwise, both the structures are same. And see this glycosidic linkage. This is a beta D galactose because the OH is above, and this is a beta D glucose, OH is above. Both are beta beta, but the linkage, glycosidic linkage is in 1 and 4 carbons. So this OH is above and this OH is below. That's why this linkage is shown like this cross line, because this is above and this is below. So remember, this linkage can be asked for the examination. Which type of glycosidic linkage, or write the glycosidic linkage in case of lactose, write the glycosidic linkage in case of maltose. So these questions can be asked for the examination, 1 mark. This polysaccharide is a deleted part. This will be not asked for the examination. Proteins also we discuss about this protein. What is the definition of proteins? What is mean by alpha amino acids? All the proteins are the alpha amino acids. Means proteins on hydrolysis gives alpha amino acids. There are also beta amino acids, but we are not interested in that beta amino acids. Only we are interested in alpha amino acids. Why this is alpha amino acids? Because on the alpha carbon, there is a COH. Sorry, there is NH2 group. See, if this is a CO, the next carbon is alpha, and to this carbon atom NH2 group is attached. That's why it is called as alpha amino. Amino group is attached to the alpha carbon. That's why it is called as alpha amino acids. The classification part is also completed yesterday. How the alpha amino acids are classified: acidic, basic, neutral, depending upon this R group. When the R group is COH, it is acidic. When the R group is basic, it is a basic amino acid. And when there is no acidic and amino group, it is called as neutral amino acids. Then this Zwitter ion is very important. Remember, this can be asked for the examination. What is meant by this Zwitter ion? Zwitter ion is a structure of alpha amino acid which contains both this positive and negatively charged. So the structure, we have to draw the structure and write in only one. This can be asked for only 1 mark. So don't write in detail what is a Zwitter ion, how it is formed. It is not required. Only in one line we have to write the answer and draw the structure.
Hmm. Lecture ಏನೋ ಆಗ್ತದ? Hmm.
Only draw the structure and show this Zwitter ion. Except glycine, all the amino acids are optically active. Remember, except glycine, all the amino acids are optically active. Anyone can tell me the reason of it? Manav Jori is there. I want to check. Yes, Manav, tell me why glycine is not optically active, or anyone can tell me. This glycine is the only amino acid which is not optically active. Why? Unmute yourself. Unmute yourself and tell me the answer. Yes.
So it doesn't have asymmetric carbon atom.
Why it doesn't have? Tell me the perfect answer. Because in case of glycine, the R group, see here, R group is hydrogen. R group is hydrogen. If we put here hydrogen, this hydrogen and this hydrogen will be same, and the definition of a chiral carbon atom will be changed. What is the definition of that chiral, or sorry, the definition of chiral asymmetric carbon atom? The carbon atom which should have the four different groups. But in case of glycine, if we put hydrogen here, this hydrogen and this hydrogen will be same. So the definition of the asymmetric carbon atom will be changed. That's why glycine is the only one amino acid which is having the which do not shows optical activity. This is a L amino acid. L amino acid means when the NH2 group is left hand side, and D amino acids when this NH2 group is right hand side. But most of the amino acids these are in L configuration. Remember, that's why it is also shown here. Here, L configuration. See, NH2 is on the left hand side. This polypeptide also we discussed yesterday in the last 2-3 minutes. This is also important for the examination point of view, because many times this is asked for exam. What is mean by polypeptide? Sorry, peptide bond or peptide linkage. Peptide linkage is nothing but when two same or different amino acids. I am coming to this example. See, in this example, two different amino acids, sorry, alpha amino acids are shown. This is one amino acid is glycine. This is another amino acid. It is alanine. These amino acids are joined together with this linkage in the square bracket. It is shown. This CONH linkage is called as peptide bond. Water molecule it is removed because this OH clearly we can see this is a CO. This OH and one hydrogen from this NH2 it is removed, and this linkage, this CONH linkage is called as peptide bond. In case of proteins, there are more than 100 amino acids linked together and form a higher molecular weight mass. Um, for example, it is given insulin contains 51 amino acids. Means 51 amino acids are joined together, and we will get the insulin protein. Then today I will start the next part. It is the types of proteins. There are two types of proteins on the basis of their molecular shape. Molecular shape mainly globular, means these are spherical shape globular proteins. They are having the spherical shape, and these are soluble in water. For example, insulin, it is a globular protein. Egg albumin, which is white colored. Egg albumin, it is white colored. Then leguminin, these are some examples of globular proteins. And these are fibrous. The name itself suggests they have the elongated, long, rod-like shape and insoluble in water. This is soluble, this is insoluble in water. For example, keratin. Keratin is a protein which is present in hairs, nails, wool, silk, and myosin, which is present in muscles. So this classification can be asked for 1 mark or 2 marks. Write the classification of types of proteins on the molecular shape. Then we will discuss about the structure. Structure is different, and this types of proteins are different. Remember, structure is different. Structure of uh, कुणाचं mic सुरू आहे? Anyone? Yes. Structure and shape of proteins can be studied at four different levels. It is called as primary level, secondary level, tertiary level, and quaternary level. These are the four protein structures. Each level become more and more complex. For this year only, due to the COVID situation, only the primary structures of protein it is kept for the examination. The secondary structure it is not included. It is deleted. Secondary structure of proteins it is deleted. Secondary is having alpha helix structure, beta plated sheet. Tertiary structure is also deleted. The quaternary structure it is more complex structures. That's why it is deleted actually. But next year this will be for the examination. So this year only we are focusing to the primary structure of proteins. This is only for the examination. They can ask for the 2 marks. Write a note on or explain primary structure of protein, or a note on primary structure of protein. Proteins may have one or more polypeptide chains. Actually, the proteins having the more than 100 polypeptide chains. More than 100 polypeptide chains are there. So that's why the more word is used here. Each polypeptide in a protein has alpha amino acids linked with each other in a specific sequence. This sentence is very important. In each polypeptide, see here in the example, see it is a CONH. It is a polypeptide chain. This CONH is a polypeptide chain. This CONH is a polypeptide chain. This CONH is a polypeptide chain. For the particular protein, this chain of polypeptide chain of alpha amino acids, it is having the proper sequence. If the sequence is changed, then the protein will be also changed. So this sequence of alpha amino acids is very important. Here we cannot see which amino acids are linked together. Only we can see here CONH linkage. But this R dash, see, this R double dash is a different R group, and this R dash is a different R group. So the structure below, representation with amino acids. We can represent these amino acids by writing three letter symbols of the amino acid. See, ALA means this is alanine, glycine. Then this is a serine, tyrine. So we can see the sequence here. So this sequence is repeated. The terminal which is on the left hand side, it is called as a N terminal, and the terminal to the right hand side, it is called as C terminal. So in the primary structure of proteins, the sequence is very important. Only we have to write, in case of primary structure of protein, each polypeptide having the alpha amino acids linked together with a specific sequence. So for each protein, this sequence is different. Each protein, this sequence is different. So if the note it is asked for the examination, we have to write these 2-3 points and show this representation. Show this representation by structural formula and representation with alpha amino acid symbols. These are the symbols of alpha amino acids, ALA. We can write this representation by structural formula. This is not required. If you remember, this is well and good. If it is not remember, um, this representation by the structural formula, we have to write this with R dash, R double dash, R triple dash, again R dash, R double dash. Follow this sequence only. See, R dash means amino acid will be changed. This R double dash, this R group will be changed. Means totally the amino acid will be changed. See, this R dash, R dash, R double dash, R triple dash. Again, we can write here R also. R dash, R double dash, R triple dash. Means here we are having the choice to write the sequence in general. This is not for the examination, but those are interested in the structures, they can read it. Already the notes are also given. What is meant by this alpha helix structure? What is meant by this beta pleated sheet? With the diagram it is shown. the tertiary structure, quaternary structure, because this is a more complex structures. Now the next part is the denaturation of proteins. For the examination, it is very important. Uh, Also make a note there in your textbook or notes. This is very, very important. Denaturation of proteins. It is always asked for 2 marks. What is mean by denaturation of proteins, or explain denaturation of proteins, or write any 2 examples of denaturation of proteins. Denaturation means actually in this denaturation, proteins का shape change हो जाता है, in which the molecular shape of the protein changes without breaking the amide linkage or peptide bonds. Means shape of the protein, molecular shape of the protein will be changed, but the polypeptide change tool is not broken. This process is called as denaturation of proteins. It is caused due to the high temperature, acid or bases, even agitation. Agitation means you are familiar with this word. Agitation means आपने उस चेन के साथ कुछ छेड़छाड़ किए, उसको डिस्टर्ब किया, तो उसका शेप चेंज हो जाता है. It is called as agitation can disturb the specific shape of the protein. Some examples are given here. Denaturation में actually क्या होता है? Denaturation means uncoils the protein and destroyed the shape and change, and also the biological activity of the proteins is also changed. It results in disturbing the secondary, tertiary, and quaternary structure of the proteins. This is very important sentence. It results in disturbing the secondary, tertiary, quaternary structure of the proteins. It is disturbed, but primary structure, in the primary structure of proteins, these CONH bonds are not broken, but the secondary, tertiary, quaternary structure will be changed. For example, it is given boiling of egg coagulates egg white. अंड अंड जर आपण गरम केलं तर ते अंड्याचं जे पांढरं कलरचं अल्ब्युमिन असतं ते एग व्हाईट त्याचं बनतं. लक्षात घ्या, हे खूप डे टू डे लाईफ मधली एक्झाम्पल आहे. कन्व्हर्जन ऑफ मिल्क टू कर्ड. दिस इज ऑल्सो द डिनॅचरेशन ऑफ प्रोटीन्स. मिल्क मधलं जे प्रोटीन असतं त्याचं शेप चेंज होतो कर्ड मध्ये. लक्षात घ्या. देन वेन द मिल्क इज हिटेड just below the boiling point, the skin forms. असा एक पापुद्रा तयार होतो ज्यावेळेस आपण दूध उकळायला ठेवतो. दुधातलं प्रोटीन म्हणजे हीट केल्यानंतर दुधात जे प्रोटीन आहे त्याचा शेप चेंज होतो. शेप चेंज होतो आणि दुधावर साई येते. साई, त्या साईला आपण काय म्हणतो? स्किन फॉर्म्स. आणि नंतर दूध उकळायला लागल्यानंतर ते स्किन ब्रेक होते. म्हणजे ही जी स्किन तयार झालेली असते ऍक्च्युली दिस इज अ डिनॅचुरेशन ऑफ प्रोटीन्स. लक्षात घ्या, दिस इज ड्यू टू द मिल्क प्रोटीन कोअग्युलेटिंग. अजून एक एक्झाम्पल मी सांगतो, याच्यामध्ये दिलेलं नाहीये. मीठ. मीठ म्हणजे मटन किंवा चिकन. इफ इट इज हिटेड, त्याला जर आपण गरम केलं तर ते श्रिंक होतं बघा, श्रिंक. This is because the protein which is present in the meat, it becomes, it's change its original shape and it becomes shrink. म्हणजे ते असं आकुंचन पावतं किंवा ते असं कॉन्ट्रॅक्ट होतं बघा. हे these are the examples of denaturation of proteins. So remember, this is very important for the short note purpose. एक्झाम्पल्स तुम्हाला अजून जे एक्झाम्पल्स आठवत आहेत तुम्ही अजून चेक करा. एक्झाम्पल्स गुगलवर भरपूर एक्झाम्पल्स आहेत. बट दिस एक से एक्झाम्पल लक्षात ठेवा. म्हणजे जिथं जिथं प्रोटीन्स आहे, ज्या ज्या गोष्टीमध्ये प्रोटीन्स आहे तिथं जर आपण त्याला हीट केलं किंवा त्याला ऍसिडची ट्रीटमेंट दिली, इफ इट इज ट्रीटेड विथ बॅस, किंवा त्याला असं आपण फिरवलं किंवा रोटेट केलं तरी त्याचा शेप चेंज होतो. दिस चेंज इन शेप, इट इज कॉल्ड एज डिनॅचुरेशन ऑफ प्रोटीन्स. हे खूप सोपं आहे. हे मुलं सोडून देतात. मागच्या 1-2 वर्षापूर्वी याच्यावर प्रश्न आला होता, मुलांनी लिहिलं नव्हतं, म्हणून मी एवढं डिटेलमध्ये सांगतोय की हे खूप सोपं आहे. लगेच तुम्हाला याचे मार्क सगळे मिळतात. देन enzymes. I am coming to the next point. Enzymes can be asked for 1 mark because the detail information of the enzymes is not given. Enzymes are, what is mean by enzymes? These are biological catalysts. In chemistry, we are using the different catalysts for increase the rate of reaction. Like these enzymes are also the biological catalysts which increase the rate of biochemical reactions in living organisms. For example, it is given enzymes helps in digestion of food, then absorption of certain molecules producing energy. All the enzymes are globular proteins, remember, means all the enzymes are spherical shape, and these are the soluble in water. These enzymes are very specific for a particular substrate in a particular reaction. This enzyme catalysis, it is shown here. See, this is active site of the enzyme. This is a substrate. This is a enzyme substrate complex is formed, and this is a product. See, this is a product means this is a enzyme. This is a enzyme means this is a catalyst. This is a substrate means this is a reactant. So the reactant, it is reacts and this product is formed. So this is for information. It is shown how the enzymes catalysis reaction takes place. It is not required to draw the diagrams, or no any question will be asked to on these diagrams. This is for only information purpose. It is shown. This is not asked. No any question will be asked on it. Remember to draw the diagrams. So example, they can ask the example also. Insulin secreted by the pancreas control the blood sugar level. Mechanism, it is shown here. Industrial application of enzyme catalysis. Conversion of glucose to fructose using glucose isomerase. This can be asked for 1 mark. Conversion of glucose to fructose using the enzyme glucose isomerase. Manufacturing of new antibodies using penicillin G. It is enzyme it is used. This is acylase enzyme. These are the names of the. They can ask the name of the write the name of the enzyme which is used for the manufacturing of new antibodies using penicillin. So this can be asked for 1 mark. Remember, don't neglect these enzymes. 1 mark question can be asked on this acylase, or 1 mark question will be asked on what is this insulin, what is the function of this insulin. So only 1 mark question will be asked on these enzymes. No any 2 marks question will be asked. Then this vitamins, it is not included in the syllabus. Vitamins, this information I have given for the students which are who preparing for the NEET exam. This vitamins is not included in the syllabus, but in the previous syllabus, the vitamins syllabus was there. That's why I included these vitamins here. What are the types of vitamins? How the then classification of vitamins, water soluble vitamins, fat insoluble. So this is very interesting. Vitamin part is there. Those students who are interested to read this, some biology students are also there. So I have prepared this notes for the students who are preparing for the competitive exam, because in the NCRT syllabus it is given, but in the state board syllabus it is not given. So read this vitamins, deficiency of vitamins, कौन से deficiency से कौन सा disease हो जाता है. This is this question it is asked for the NEET exam. See, the vitamin, different vitamins are shown. This is for general knowledge also. We can read the vitamins for general knowledge. Vitamin A, which is present in fish liver oil, then carrots, butter, milk. इसकी deficiency अगर हो गई तो कौन सा disease हो जाता है. देखो, night blindness, then uh, hardening of corona of eye, then vitamin B1, then vitamin B2, vitamin. These are the different types of vitamins. See, vitamin A, B1, B2, B6, B12. Then vitamin C is also called as ascorbic acid. Then vitamin D, vitamin E, vitamin K. All these vitamins are shown. Its sources are also shown. This table is very important. Then I am coming to the next part. It is the last part of the chapter. It is nucleic acids. Transfer of genetic information to the new cells. It is done by the nucleic acids. There are mainly two types of nucleic acids. One is RNA. It is called as ribonucleic acids, which is found in the fluid of living cells, cytoplasm. And next nucleic acid is deoxyribose nucleic acid. It is called as DNA and RNA. These are the short forms of nucleic acids, RNA and DNA. What is the role of this nucleic acid? To transfer the genetic information. Remember, to transfer the genetic information to new cells. It is the function of this nucleic acid. So this DNAs, these are found in the nuclei of the living cells. Remember, these are found in the nucleus, and these RNA, which are found in the fluid of living cells, cytoplasm. Then I'm coming to the next part. It is nucleotides. Nucleic acids are unbranched polymers of repeating monomers called as nucleotides. जे न्यूक्लियोटाइड्स पासून न्यूक्लिक ऍसिड बनतं, लक्षात घ्या. So nucleic acid nucleotides are act as monomers, and nucleic acids, these are the unbranched polymers. They consist of three components. One is monosaccharides, then nitrogen containing base, and phosphate group. These structures are very important. Remember, these structures can be asked for 1 mark. How the, or write a note on nucleotides also. So nucleotides, they consist of three components. One is monosaccharide should be there. Then nitrogen containing base should be there, and phosphate group should be there. Nucleotides, both the nucleotides RNA and DNA contains 5-membered monosaccharides. It is furanose. Often it is called as a sugar component. In this diagram it is shown. See, this is a 5-membered monosaccharide, furanose it is shown. See here. In RNA sugar, the unit is D-ribose. In DNA, it is 2. It is shown here. See, D-ribose, which is present in RNA, and this is a, in DNA, it is a 2-deoxy D-ribose, which is present in DNA. See, it is also written here. Uracil occurs only in RNA, while thymine occurs only in DNA. Remember this. This can be asked for the examination. Uracil, which is occurs only in RNA, while thymine, which occurs only in DNA. 99%, 99% the structure will be not asked for the exam. Remember, 99%. I am talking about the examination point of view. 99% these structures are not asked, but 1% there is a possibility. But actually, it is not required from the student point of view to draw the structure. Student only should know uh, what is meant by nucleotides. Nucleotides contains what? Nucleotides means they contains the monosaccharide, nitrogen, phosphate. This is important. So they can frame a question on it. Which are the three main? Write the three main components of the nucleotides, or write the components of the nucleotides. Such question will be asked for 1 mark also. So this is a structure of D-ribose, and this is a structure of deoxyribose. See the difference between these two structures. OH here, oxy deoxyribose means this OH is no OH at the deoxy. That's why it is called as deoxy D-ribose, because this OH on the second carbon is removed. That's why it is called as a deoxy. And in the D-ribose, OH is there on the first carbon, second carbon OH, third carbon OH, and fourth carbon CH2OH is there. So both the structures are actually same, only on the second carbon OH is not there. That's why it is called as deoxyribose, which is present in DNA. Then nucleotides are different. This is nucleotides are different and nucleosides are different. Then I am coming to the nucleosides. The nucleosides contains the pentose sugar, furanose, and nitrogen base. There are total 5 nitrogen containing base which are present in the nucleic acid. These structures will be not asked for the examination. Cytosine, this is uracil, this is thymine, which contains the nitrogen base also. This is adenine, this is guanine. These are the bases which are present in nucleic acids. These are called as nucleotides. Then the structure of nucleic acids. This is the structure of nucleoside, and this is B is the structure of nucleoside and nucleotide. Don't get confused between nucleotides and nucleosides. So B is here structure. B is it is a nucleotide, and this is A structure is a nucleoside. These structures are asked for the examination. So make a practice today. Make a practice of these structures. This base in the nucleoside. The base is given, and other structure is very easy. Only instead of this OH group here, phosphate group is there. Both the structures are actually the same, but only in the structure of this nucleotide, instead of this OH group, there is a phosphate group is there. These structures are not asked for the examination. This will be not asked for the exam. This is deleted actually part. So this chapter is completed here. DNA double helix structure. This is the Watson and Crick model of DNA. This DNA structure, it is not asked for the examination, but this is a double helix structure. Double helix structure, James Watson and Crick in 1953, they discovered this structure of DNA. It is verified by the microscopy. And these are the some salient features of these structures are shown. DNA consists of two polynucleotide strands, which are shown here. This is one nucleotide strand, and this is another nucleotide strands. With a right-handed double helix, two strands run in opposite direction. Sugar phosphate backbone. The double helix, which is stabilized by hydrogen bonding bases in two